present here. I am Abhimanyu from Proteus, the Biology Club of IISER Tirunelveli, and I shall be your host today. Proteus is delighted to welcome you all to our 8th talk in RHETOR 3.0. RHETOR started as a mini symposium with guest speakers worldwide, indulging our audience in various exciting developments in the scientific arena. We are excited to conduct RHETOR with full enthusiasm and at a larger scale this time. The theme of RHETOR 3.0 is etc., a timeline of biological manipulations. We will explore the concept of hierarchy of the biological organization from molecular biology to astrobiology and leave it open-ended for the future of biology to decide upon the idea. The topic for our 8th talk will be training machines and humans to democratize data-driven biology, and our speaker for the day is Dr. Arjun Krishnan. He is an associate professor of the Biomedical Informatics Department and Center for Health AI at the University of Colorado in the US. We warmly welcome you, sir. Dr. Arjun
Thank
Krishnan obtained his BTech in Industrial Biotechnology from AC College of Technology in Chennai. He completed his PhD in genetics, computational biology, and bioinformatics from Virginia Bioinformatics Institute, US, where he also worked as a postdoctoral research associate. He was also a postdoctoral research associate and associate research scholar at Princeton University. Before joining the University of Colorado, he worked as an assistant professor at Michigan State University. His research lab, the Christian Lab, develops and applies computational data-driven approaches for resolving, understanding, and tackling the heterogeneity of complex traits and diseases. In addition to biomedical data science and machine learning, he is passionate about open science, research training, and creating diverse and inclusive learning environments. We are honored to have you here today, sir. Before we begin the talk, I request the audience to turn off their cameras and mute their microphones. Please reserve your questions until the end of the talk and we'll have a fruitful interactive session. I'd like to pass the virtual microphone over to Dr. Arjun Krishnan now. Thank you.
Thank you. Thanks a lot. This is a wonderful opportunity for me to, meet all of you, albeit virtually. And it's a fantastic effort that all of you folks are doing. And I would really want to congratulate you on, organizing this year after year and doing a great job at it. So maybe slightly in contrast to what Abhimanyu suggested, I would actually like to see some of you at least so that I get some feedback, because otherwise it's like blank screens. So I'm not really sure. I wouldn't be sure. So if some of you would volunteer to turn on your videos, that'd be great. And then sometimes I would ask some kind of generally whether it'll be great for me to ask some of you whether I'm going okay, and and if you can give me a nod or a thumbs up or just shout out an answer, I would really appreciate it. Okay. All right. So awesome. Thank you everyone for turning on the cameras. That's great. All right. So let me, share my screen first, and then we'll go from there. Right. Entire screen tab. Okay. So let's see. Yeah, I'm assuming you can see my screen, right, and my presentation, which is going to load very soon. All right. Okay.
Yes, we are able to. Yes.
Super. All right. Okay. So like Abhimanyu said, I'm Arjun. You can just call me Arjun. I'm an associate professor here at University of Colorado. I moved here very recently. And the best way to introduce myself in a different way is that I'm a Jurassic Park kid. Okay. I watched there was this movie in Madurai in a small town called Nagamalai in 1993, and I was totally hooked. Okay, because it was amazing as a child to basically see even this possibility that someone could extract frozen blood from a mosquito in an amber stone, extract DNA and make an entire organism. And right then and there, I surely remember that it would be wonderful to get some opportunity in my life to even study genetics, leave alone work on it. But it's been an amazing journey from that point on. And like Abhimanyu said, I did my undergrad at Anna University. That's where I first discovered this area called computational biology, because up to that point, even during undergrad, I was going to labs and doing, PCRs and running agarose gels and so forth. And I was no good at, experiments in the laboratory. But then I met this PhD student who was working in this area called bioinformatics. And then he was basically saying that he's doing biology research, but sitting in front of a computer. And I thought, okay, what is this? I have no idea what he's talking about. So I basically followed him up, started talking to him, and then really discovered this whole area of writing code and thinking about algorithms and then using that to solve important problems in biology. So that led me to grad school here in the US. And then I completed that at Virginia Tech, where I studied how plants respond to environmental stress. And then the work that I was doing was to say, how can you take advantage of data that is available to all of us in the public domain, but using that to figure out which are critical genes in the genome that make these plants respond well or not to various stresses like very less water or very high heat and so on. Okay. And then I went to do my postdoc, and then during my postdoc, I made this big shift from studying things in plants to studying things in humans, which is what we do in my group right now. And there, one of the things that I started doing was to say, okay, how can you use, again, publicly available data to figure out how genes interact with each other in specific tissues in the human body. So, for example, all of us know that our genome is almost exactly the same in all the cells in our body, but we also know that the brain cell is very different from the cells in the heart to cells in the kidney and cells in the lungs, right? So this is because not because the genes are very different, but some of these genes are turned on and off to different levels, but also the fact that these genes have very different types of interactions with each other. So, for example, the same gene will have very different interaction partners in different tissues and cell types, and we sort of help figure this out. Right now, this is my research group. I started my position at MSU, and then now I continue at University of Colorado. And this is a terrific group of people, and I really want to give a huge shout out to them. These are all postdocs, graduate students, programmers, but also undergraduate students and high school students. Okay, these are people like you, for example. The people here, they're all undergrad students and high school students who came to our group and then started working with our in our research program for more than 2-3 years. Some of them have authored papers. All of them have gone to conferences, presented their work, and all that stuff. So I'm very proud of this group of people. Okay, so as a group, we think about three different aspects of complex diseases, and I want to lay that out right now, and then talk about some of them as we go forward. The first one is that even if you take every single person who is diagnosed with a particular disease, let's say diabetes, right? It is not true that every single person who's diagnosed with diabetes is identical. There are subgroups of people who have this disease, but for very different genetic reasons, because they have very different mutations in their DNA, or even when they come to the clinic, they present the disease very differently. When the doctor makes all these measurements and tests, they will show readings that are very different from others who have the same disease. Okay? But still, we sort of combine them as a single disease, which is actually a problem in both diagnosis and treatment. So one of the things that we are very interested in is trying to understand, how do you figure out if subgroups of people who have this disease, but for very different genetic reasons, and also have very different clinical presentations. The other problem that we work on is, I'm going to talk about this down the line, so I'll keep this brief, is to sort of lay some groundwork to understand how and why diseases vary across the age and between sexes, males and females, right? So this is why, for example, why is autism way more frequent in boys than girls? And it, of course, occurs very early in development. And if you think of something like arthritis or rheumatoid arthritis, it is way more common in women than men, and it occurs sometime during 40 to 50 years of age, right? It sort of starts happening. So we want to understand why this is all important. Now, the last area of focus for us is to see, I think all of us may have some level of familiarity that a lot of work that is done in biomedicine to study human diseases, many experiments to understand the underlying mechanisms are conducted in model organisms like mouse, fly, fish, right, and worm. So it is really important to be able to translate any data that we generate in one organism and sort of move it to another organism to figure out how you can do this comparative analysis across species. So we are very interested in developing methods to do that. Okay, all right. So to do all these things, what, like Abhimanyu mentioned this, I mentioned this a little earlier, which is that we want to take advantage of data that is publicly available. What this means is that any one of us can right now access all the data that I'm going to talk about. Okay, it's not generated by some lab that shared it only with us. Okay, all right. So the types of data are, for example, on the top left, I'm showing you a type of data called omics. And these are all, technologies that were developed in the past 20 to 25 years. And these are technologies that help us essentially measure let's say, the activity of all the genes in the genome in a biological sample in one shot. Okay. There are also technologies that allow us to figure out if you take a single protein, you can figure out what are all the locations along the genome where this protein binds, again in a biological sample, again in one shot. So these are super fast, high throughput technologies, and researchers have been using these technologies for the past 25 years. The cool thing is that once they conduct an experiment and generate this data, these researchers have also been depositing these data to a public database. So which means that from past 20 years, data generated by researchers from across the world, okay, have all been deposited in a common database, which we can now access and start using to figure out interesting things. Okay. The same goes for the data that I'm showing you on the top right. This is all, mutations and genes that are associated with diseases. The typical, studies that people conduct is called genome-wide association study, in case you're interested in looking up. But these are also the kinds of things that, yeah, for example, this data also includes information about the individuals from which we got the DNA and we found out these mutations. And this information is sort of, buried inside health records. Okay. A good way to think about health records is, for example, when we go to the doctor, typically someone is typing, either doctor is typing or, some other nurse practitioner is typing, or we are filling forms about ourselves, about our medical history and all that stuff, right? All that together is the electronic health record. The data at the top middle of my slide, these are all data that is essentially, you can think of this as knowledge. Okay, knowledge meaning that somebody has done careful small scale experiments and they have figured out the roles of that various genes and molecules play, and they have described this in the literature, or some of it has been curated in databases. And this data comes from both humans and also many model organisms, like I said, worm, fly, yeast, fish, and so on. So we deal with all these types of data, and the way we sort of take advantage of all of them is to build machine learning models. These models are predictive models, which means that we are not only building some model to try and understand what's going on right now, but we also always want to be able to predict something about the future. What I mean is that maybe a novel role that this particular gene plays, or maybe what is the disease status of this specific individual, and so on. So we always want to predict something about the future. Okay. Hopefully that makes sense. Okay. Okay. Right. So let me talk about, to give you a sense of what I mean by, data and all that stuff. Let me give you a sense. So here's a table I put together back in 2020. This is already old. Okay. So if you sort of take the 14 most common types of experiments in this high throughput, omics type experiments, you can already get access to almost 26,000, 800 data sets. And together they constitute about 1.3 million genomic profiles. Okay. I would like you for a second to think about this, right? This is all data essentially from hundreds of tissues in the human body, generated from people around the world. Okay. And these are all samples that come from various genetics factors, environmental factors, physiological factors, clinical factors. And both clinical and basic scientists have been studying these samples, and they've been depositing all this data in one single place. Isn't that amazing, right? So now, if you sort of expand this data to data that comes from many other organisms like mouse, fly, worm, and fish, this goes all the way up to 2 million profiles. Each of these profiles contains some information about all the genes or all the locations in the genome, and there are 2 million like this. Okay, so now wouldn't it be awesome if we can actually use all this stuff, right? So in fact, lots of researchers, including my group and myself, we have shown that these publicly available data are super valuable, because what you can do is to, even without generating any new data or doing any new experiments, you can just go to existing data and then generate brand new biological or medical hypotheses, right? Or then you can say, you can go back, and if that data is available, you can even go back and reanalyze the data to figure out if the conclusions drawn by the scientists are actually correct. The third thing that you can do is to take data from multiple studies, bring them together, and do some integrative analysis. The fourth is you can sort of say, I'm going to generate some new data in my lab, but I want to figure out how much support there is in what I find in existing data sets. Right? And the fifth is to meet urgent scientific needs. For example, in the context of COVID, when the pandemic hit for the first time, we had no idea about this virus or what's going to happen in this infectious disease. So one of the first things that scientists did was to look at data that is already out there, but for related viruses and related infectious diseases, to sort of try and understand what's happening, and then maybe use that to sort of find maybe potential drugs, and then start the journey from there. And then later on, we were able to generate new data specifically for COVID and so forth. Right. Okay. Now, I told you that many of us have already used all these public data sets, right? But it is not true that every biologist is able to use all this data. That would be awesome, but this data is actually acutely underused. This is because there are two major problems with these biomedical data, and they have to do with metadata. Okay? Metadata is the description of data, okay? And then there are two problems here. The first is that metadata is unstructured, which means that all the descriptions that scientists write about what the sample is written in plain English text so that other scientists can read and understand them potentially, right? But think about I already told you that there are millions of samples. So you can't expect humans to read each of these descriptions and figure out what the sample is. So we need to bring a lot of structure to this description, but it's currently not available. The second part is that of missing metadata. All right. So this is the case where scientists have written descriptions, but this description is missing key pieces of information about the original sample. So I'm going to tell you about that too. Okay. All right. So let me first begin with unstructured metadata. Okay. If you take any sample for which there is data in the public domain and look at its description, the description will typically look like what I'm showing you on the top right of this particular slide. It is just a block of text. There are a bunch of words. Okay, somebody who's an expert needs to be reading this very, very carefully and then trying to understand where the sample actually came from. Let's say you want to figure out which tissue in the body the sample came from. You really have to read this carefully. Okay. Second, scientists are also, although we are known for bringing a lot of standardization to describe what's happening around us, we still end up using slightly different terms to refer to the same thing. For example, you will see that somebody will say myocardial infarction, or somebody else will say heart attack. They mean the same thing. Okay, so if you're just going to do Google search or just do any search based methods, you would just have to know all these various ways in which people are researchers are referring to the same thing. The third is, all of you, I will encourage you to read a sentence like this, right? It says, blood was drawn from patients with kidney cancer. Macrophages were isolated and they were used for transcriptome profiling. Okay. Now, there are names of 3 different tissues in the body: blood, kidney, and macrophage. But we as humans know that the transcriptome profile was generated from macrophages. But if you were just doing keyword searches, you'll hit blood, you'll hit kidney, and you'll hit macrophages. So you won't know which is actually the source. Fourth is, there are many cases where scientists write, let's say, the name of the disease that they are interested in studying, but they don't write the tissue from which the sample was collected to study this particular disease, right? So for example, somebody will say psoriasis, but they would have completely left out the word skin from the entire description. But we know that psoriasis is a skin disease, and hence we can potentially infer that the sample is from skin. The same for cerebral palsy. And the last issue is things that we encounter every single day. It's like just spelling mistakes, right? So like cardiomyocytes, just spelled differently. So these are all problems. So a graduate student, Nat Hawkins, and an undergraduate student, Mark Moldaver, essentially started working on this problem, and they developed a machine learning method that's based on natural language processing. This is the computer science field that essentially looks at language that is written for other humans and parses out information from it in some clever way. Okay. The underlying basis for this entire effort is called, word embedding. And this is a technique that we borrowed from the field of computer science. And it's burgeoning right now. And then there are many, many sophisticated models. But the underlying idea is pretty straightforward. The idea is that you can build a neural network model that takes massive amounts of text data. What I mean in this, take all the data, all the text in Wikipedia, all the text in Google News, or all the text in every blog post that has ever been written, right? All the text in every scientific paper that has ever been written. So you can essentially take all this text data and then feed this to a neural network. And what the network will allow you to do is to now turn every word in this entire data set into a set of numbers. Okay. Now, some of you might be able to see what is the advantage of doing this, right? Words are just characters next to each other, but many of the algorithms and statistical methods that all of us have studied, even in classes, we know that the input for all of them needs to be numbers, right? We can't say, okay, here's a word, do some statistical analysis on it, right? So this is where this approach becomes very powerful. So we take an existing neural network model that can do this for us, turn every single word into numbers, and then we're going to use this for tackling the unstructured metadata problem. So the way we are going to do this is to first curate a bunch of samples for which we manually read the descriptions and make sure which tissue it actually comes from. Okay. Once you have all these examples, we do some pre-processing, and then what we do is to turn the description of every single sample into a set of numbers. The way we do this is that every sample's description has many words in it, right? So we know how to turn every word into a set of numbers. So we take the set of numbers representation for every word in the description and simply average them together. Okay? That's how you get a numerical representation. For example, sample A is 28016, 164469. This set of numbers we get from the description of the sample, the words that we use to describe the sample. Okay? Once you have this data, what we then do is to train machine learning models to recognize samples that come from one tissue and set them apart from samples that come from completely unrelated tissue. And then we do this for every tissue: heart, lung, kidney, liver, and so on. Once we have all these models, then what you can do is that somebody can just type up some descriptions or take an existing description of some sample. We have no idea which tissue it came from. Do the same pre-processing, turn them into numbers, and then feed them as input to our pre-trained machine learning models to then get predictions. The prediction is, for this new sample, I have no idea which tissue it came from, now gets a probability that it belongs to heart, that it belongs to brain, that it actually came from kidney, and so on. Okay? And you can look at these probabilities to decide which tissue it probably came from. Right? Hopefully this makes sense. Okay, so to give you a sense of, I really want to, these are things I really want to, speak about especially when I'm talking to students who are thinking about data and all that stuff, right? So here, I'm showing you the performance of this task of predicting which tissue the sample came from based on its description, but using a previous method called Metasra. Okay? Metasra is this method here, and then I'm showing its performance along this axis, and the axis is average precision. It means that if the precision is 0.5, it means that if Metasra says here are 100 samples related to the liver, 50 of them are likely to be correct. That's what 0.5 means. Okay. And one thing you will see that if you're familiar with box plot, Metasra has a wide range of performance. Okay. There are some tissues for which this previous method does really well. There are other methods for other tissues for which it does really poorly. For most of them, it gets about half the things correct. So in this context, here is the performance of our method. Okay, we get on an average 75% correct, which means that if our method says 100 samples are related to liver, 75 of them are likely to be correct, right? And our range of variation is pretty small, which means that most of the time we do better than Metasra. Okay. Now, whenever somebody shows you plots like this, you should not believe that, okay, this is a super method, this is working better than previous methods, and we are done. Okay, it's never the case. It is really important to dig into these results and see where is it really doing better in every single tissue, right? So for instance, here is a plot that I'm showing you where the previous method is along the x-axis, our method is on the y-axis, and every single point is a tissue. Okay? If the point is above the diagonal on the left, it means that our method is better. Okay. So here I'm showing you a bunch of cases where our method is almost equal to the previous method, or it's better, right? But this is only a subset of tissues. So if I show you more data about what's going on underneath, I want you to look at the plot on the top left of my slide. Okay, these points here. These are cases where we have very little training data to feed the machine learning model. In these cases, there are a considerable number of tissues for which the previous method is way better than ours. In fact, we perform very poorly, but the previous method does really well. Okay. If you look at the tissues in the top right, you'll see that these are all tissues for which we have a large amount of training data, because of which we perform really well, way better than the previous method consistently. Okay. So now, here is a question for all of you. Okay. If there are two methods that use completely different ideas to solve the same problem, and I tell you that one of them is slightly better than the other, what is the next step?
So you generally
The better one.
oh, I'm sorry. Please go ahead.
No, first Anagha, and then maybe Prabin, who.
Yeah, so generally, depending on the amount of training data you have, you would select which model to choose. Although I
Nice.
was a little confused regarding the fact that as the training data changes, why isn't Meta SRA also performing much better? Because
Very,
generally, every ML algorithm performs better over training
very
data.
nice. Okay, that's very good. Okay, anyone else wants to comment? I'll come back and answer and ask very good questions. Any other thoughts? What do you do next?
Maybe try and see if the training data is low, and if the new method is able to give a at least a bit good results rather than the very good result given by Metasra. And when the
Right.
training data is very high, actually our method, the new method, is giving better results. But when
Right.
we compare Metasra, if it is giving at least a bit good
Mm-hmm.
results, then we can go with Metasra also, right?
Yeah. Okay. So you agree with Anaga. Okay. All right.
Yeah.
Others? Anyone else? Okay. I'm sure you're also thinking about something that I'm going to tell you. Okay. So first, very good question, Zara. So Metasra is not a machine learning method. So I should have maybe clarified that. So it is not using training data to train models. It basically uses our understanding of how tissues are related to each other, and then looks at one sample at a time, and there is no training. So that is why it's not improving with training. Okay, so that's a great question. Okay, so some of the ideas is that just to choose the one that's better, especially depending on the tissue and depending on how much training data there is. There is another solution, which I'm sure you're thinking about, which is just combine the predictions of both methods for all tissues. Okay, so here is the combination. Okay, so this combination, as you can see, it performs way better than either of these two methods, our method and the previous method. And I'm showing you the performance of the new combination on the y-axis and the previous Metasra's performance on the x-axis. And across every single, any type of training data that you have, you will see that most of the points are above the diagonal. Okay, so when there are multiple methods, they use slightly different approaches to solve the same problem. Invariably, combining the predictions for every single case gives you almost always better performance than either of these original set of methods. It can be 2, it can be 3, or whatever. Okay, all right. So again, in machine learning, it's very important to not be satisfied with just performance, okay, or accuracy, because these models, I'm going to tell you about this a little later in the talk, these models are very good at capturing hidden patterns, even if they are nonsense patterns. Okay, so it's really important to go back and figure out whether these models are capturing something that's biologically meaningful. So the way we do that is, remember, we have one model for every tissue in the human body. So what we did was to take these models that were trained and just compare them to each other. And I'm showing you the comparison as a plot where every single point is a machine learning model for different tissues. And the shape of the point or the color of the point, it depends on which organ system the tissue comes from. So what we see is that invariably, tissues that come from the same organ system end up having very similar machine learning models. Okay? That's how we know that these models are at least capturing a certain level of biologically meaningful data, words in the descriptions. All right. So to conclude this part of the talk, we love doing the following, which is that we actually do not believe in developing an algorithm and describing them in some paper so that others can read it. We actually want to implement this as software so that researchers can immediately use what we have developed and then also build on top of it. This software is open source, so which means that the underlying code is available. any of you can go and fork this code and modify it and even suggest changes for us. If you want to improve this code, it's already there. We've also written utilities so that you can use our existing pre-trained machine learning models or even build new models based on this code. So if you're interested in biomedical NLP or machine learning, you should definitely check this out. Okay. All right. So we are doing a bunch of stuff going forward, trying better word embedding methods, trying to improve the way we can explain machine learning models. Not trying this not just for the tissue from which the sample came from, but also the disease or the environment and so forth. I told you this, we did this work just with human data, but we want to do this with data from other organisms. And we also want to build a web server where we sort of run this model on all existing data, make annotations, and make these annotations available for the entire community, so that someone can come and use it like Google, right? So they can say heart, and we should be able to provide every single sample that was ever conducted with heart samples, right? And so on. Okay, so this is what we are working on right now. Okay, so to remind you, what I've told you up to now is we are just starting to develop methods to tackle the problem of unstructured metadata. The second thing that I'm going to tell you is about missing metadata. Okay, and I'm hoping that the host will also give me some heads up on the amount of time that I have, right? When it's about 5 minutes out, just let me know. Okay. All right.
Yes,
So
sure.
okay, got it. So the missing metadata problem is when we encountered this a lot when we started thinking about sex and age differences. Okay. So now, how many of you have heard the term precision medicine or personalized medicine, at least at a high level? Okay. Okay, great. Some of you are raising your hands. Okay, that's awesome. Great. So we know that precision medicine is this idea that somehow we can tailor our diagnosis and treatment for one single patient who comes into the clinic, right? Now, this is like this big goal of biomedicine, but there's a huge contrast. Right now, it is actually pathetic that we as a scientific community, we lack very fundamental understanding about genes and molecular mechanisms for more than half of humans. You know who I'm talking about? Women. Okay. It's like 51% of our population is women, but our understanding of disease mechanisms, basic physiology, diagnosis, treatment, all of this is very poor in women. So for instance, if let's think about heart attack, right? If men get heart attack, let's say if I get a heart attack sometime down the line, I will have a sharp shooting pain near my chest, and I will know something is wrong, and I know how to go to the doctor. However, when women get heart attacks, they have pain they have diffuse pain in different parts of the body. They'll get a shoulder pain, they'll get pain in their back, lower back, they might even get pain in their thigh. So what happens is women will say, okay, there's some pain, I'm going to try something, maybe apply some balm, wait for a week, the pain won't go away, and then they will go to the doctor. But by that time, a lot of damage has already been done. Okay. So although the rate at which heart attack happens is the same across males and females, heart attack is more fatal in females. Okay. Is that clear? Okay. So this is diagnosis, right? What about treatment? Most drugs that are out there in the market for treating all these diseases have almost never been tested in females. Isn't that ridiculous? It's like half the population, and these drugs have never been tested in women. So which means that doctors treat women and females as slightly smaller males. That's it. So all they do is to reduce the dose a little bit and give it to females. This is why if you look at a people who are having these adverse side effects of all the drugs, invariably these side effects are way higher in females because they have never been tested in the first place. We also lack very poor understanding of fundamental mechanisms and genetics and all stuff. So one of the goals for us is to really try and fix this and help the biomedical community go forward in this direction. Okay. So the way we are going to do this is to say, we're going to go back to some of the method work that we've already done. We have done some work where we can say, if you take public data sets like gene expression data, this is from thousands of studies, we already know how to take these studies about which genes are active in which conditions, and then use that to predict gene interaction networks in various tissues in the human body. You should think of each network as a social network of genes. Which genes are friends with which other genes, but in specific tissues in the body. We also know how to use these networks to study complex diseases. Like I've done a bunch of work with studying using the brain-specific gene interaction network to study autism. For example, genes that are associated with autism. How do these genes specifically interact with each other? And what is their relationship to brain development and so on? Okay. So we thought, okay, we know how to do this stuff. We are simply going to take this existing data, now group them by sex and age, depending on the description, and then build not only tissue-specific gene networks, but also sex and age-specific gene networks, and then use that to study sex and age-specific disease mechanisms. Okay, is this logic hopefully makes sense to you? Okay. However, when we started trying this, we hit a roadblock right at the beginning. Okay, this first part, because remember I told you we have to try to understand the samples based on the descriptions provided by the scientists, right? Turns out that 60% or 70% of this data is completely missing information about the sex of the individual who gave the sample or the age of the individual from which the sample was collected. Okay, this is astounding. Okay, so this information is not even there in the text. So how do you can't do NLP, right? Because this information is missing. So now what this work was actually led by an amazing graduate student called Kayla Johnson. She's actually defending in a week. So what Kayla has been doing is to say, okay, we have all this data that's available. She's going to use the data itself, the molecular data that the researchers have already measured from all these samples, and use that to predict which sex the sample came from and which age group the sample came from. And then use that data to separate them into sex and age groups, and then build model networks. Okay. So just to remind you, we know how to do the latter part, but we don't know how to do the former part. Okay? And this is what Kayla has been working on, to develop a machine learning model that uses data to predict metadata. All right. So some of you know that machine learning models require training data. So it turns out that the data that was already there was not good enough. So for example, the largest study that had enough data, rather a reasonable amount of data, is called GTEx. And they had around 16,000 samples, and 2/3 were males, 1/3 were females. And then the ages was not spread across, the whole human lifespan. So what Kayla did was to go back to these data sets, these public data sets, and then curate almost 613 data sets, which amounted to 30,000 samples, almost equally split between males and females from 2 different technologies, and also spread across the entire human lifespan from fetus all the way to elderly, which is greater than 80 years of age. Okay, so we have data spread across the age groups and between males and females from 2 different technologies. So this is a tremendous amount of data. So some of you might have already guessed that you can actually look at, because let's say we know the activity of all the genes in the genome, you can look at the activity of even single genes, like a gene on the X chromosome or a gene on the Y chromosome. And based on its activity, you can with a very high accuracy, predict whether the sample from which the person who gave the sample was actually a female or a male. Okay, because we know that in females, the activity of the gene on the X chromosome will be higher. In males, the activity of the gene on the Y chromosome will be higher, right? So for example, this gene on the right is a gene on the Y chromosome, this gene on the left is a gene on the X chromosome. And just based on its activity, you can predict female or male. So this is actually not a hard thing to do. Okay. Predicting age group is, however, a little tricky. So what Kayla did was to divide all these samples into 11 age groups. I'm going to tell you what she did for just the fetus, but you should imagine that we repeated this for every age group, right? So let's say we want to predict whether a sample comes from fetus or from some older age group. So we set all the samples in fetus as positive examples, all the other samples from all the other age groups as negative examples, and then train a machine learning model. Okay, I'm showing you a very bare bone version of this model, okay, so that we can discuss what's happening underneath. This model takes the expression of every single gene in the genome, and those are all the X values, X1, X2, X3, and so on. X1 is the expression of gene 1. Okay, and what the model is supposed to do is to, based on the expression of all these genes, predict whether the sample came from fetus or from some other age group. Okay, now the way the model is trained, the model is trying to figure out which genes are important and by how much. And the way it does this is by us supplying the model with many examples of gene expression levels, and also telling the model whether it comes from the positive group or the negative group. Once we do this, the model figures out what is the strength and the direction of importance of each of these genes. This is like tens of thousands of genes, right? So this is really cool because it tells us for every single gene, is it really important for fetal stage, and is the importance based on high expression or low expression? Okay. Now, here is a way to look at the performance. This is a complicated plot, but let me take a second to go through this. I'm showing you all the models that we trained along the rows of this matrix. So for example, the first row corresponds to the model that you trained to classify fetal samples from non-fetal samples. But once you train this model, what we did was to make it predict samples from all the age groups, which are all along the columns, irrespective of whether it came from fetus or not. Okay. What we hope is that if the samples really came from fetus, the model will have very high accuracy. But if the model came from any other age group, it will have low accuracy. Okay, because the model is trained to predict fetal samples. Here, it turns out that it's actually true, right? The dark square is the one that actually corresponds to fetus. So in general, we expect the diagonal to be very high, okay, or higher than all the other values. However, this also helps us. In general, this turns out to be true irrespective of whether you're looking at female samples or male samples and across technologies. However, doing this very extensive evaluation also points us all the problems with the underlying models, right? For example, here is a model that we trained to predict 20 to 35-year-olds. And across the row, you will see that you don't see any dark square. So which means the model is pretty poor across the board. And it also shows us there are samples from particular age group like infant, which seem to be high irrespective of which model you're providing as input, right? And which means that we need to go back, and this is also the stage for which we have very low training data. Okay, so such evaluations also give us a really good insights about both benefits and problems with machine learning models. Nevertheless, even though I've shown you problems, these performance is actually pretty good. To just convince you, what I'm going to show you is the performance of all the diagonal values, which we are supposed to be high. Okay, now here's a question for you. What do you think about the performance for all these squares that are right next to the diagonal? Do you expect them to be really low, or what do you expect?
It would still be expected to be around the darker colors as compared to those which are farther.
Nice. Okay. I'm assuming others are thinking along similar lines. Okay.
Yes,
Now,
sir. Yes.
okay, very good. So these are I'm going to call them the bordering ones. The ones I'm going to call them close by are next to that again. Okay, not exactly the bordering one, but the ones next to that. And then all the other ones I'm going to call distant age groups. Turns out that if you look at the performance, it is exactly what we expect. The diagonals are the highest, the bordering ones are the next highest, the close by ones are the next highest, and the distant ones are the least related to the target models. And this is true irrespective of female or male models and depending on the technology. Now, here is the cool thing, okay? We never told them, let's take the fetal model, right? We never told this model that infants are related to fetus. Remember, we just put all the samples that is not fetus into the negative bin. But even then, somehow the models understands that infants is closer to fetus and gives it a slightly higher probability than a sample that comes from very old age, right? So this is how we know that the model is learning something biologically meaningful, because it is somehow capturing the chronology of human aging. Okay. All right. So what we've been doing is to going back to this model, looking at these beta values, which tells you which gene is important for every single age group, and understanding that in terms of genetic mechanisms, molecular mechanisms, and so on. Okay. So this is just ongoing work, right? Okay. So what I've told you now is that we have a machine learning model that can actually take existing data and based on the molecular data that's recorded, figure out which sex and which age group the sample might have come from, and use that to group this into various sex and age groups. And then we're going to be building network models right now. Okay. Let me skip this. Okay, so here is the summary of what I've told you. We have developed a natural language processing method to deal with unstructured information that is present in the descriptions of these samples. And if that information is completely missing, we have developed methods to deal with missing metadata. All right. And the first method has already been published. It's out. It came out a couple of months back. And then the second one, Kayla is writing up right now. We're going to submit this soon, and you can surely expect a preprint from our group about this. All right. So in general, these are just 2 projects, but we have tens of projects that are going on. We work all over in the area of natural language processing, data representation, knowledge representation, harmonizing publicly available data, building machine learning models that are informed by biology, and also building lots of software and web tools. Like I said before, if some of you have talked to folks who publish papers, or some of you already have experience doing so, we know that paper publishing is a crazy system in science, which is actually slowing down science. Because what happens is you write a paper, you submit to a journal, and then they take about almost sometimes 6 months to 1 year, or sometimes even 2 years to complete a review process by experts in the field. This is all hidden behind a curtain, and then it gets published. But with science moving at such a fast pace, what use is something that was discovered 2 years ago or 3 years ago, right? Just slowing down science. So what we and many of us in the community do is to, when we write a manuscript and submit to a journal, we also immediately make it available to the public, the community of scientists, and anyone can read it. We also make all the data and the intermediate results of our analysis available. We write all the code and make the code available so that anybody can go and reproduce our results and check our code and do whatever we want. And we've also been developing web interfaces so that scientists who don't want to write code but still want to use our algorithms can go to a Chrome window and then type in things, and then we will run algorithms in the background and then give them results back. All right. So depending on how much time I have, I will go into another small part. So tell me, Abhimanyu.
So we are actually about to towards the end of the session, but
Okay.
we need some time for interaction. So I think maybe you can go forward and talk for at least 5 more minutes, maybe.
All right. 5 minutes sounds very
Yeah.
good. Okay. So because I think this part of the talk is also super important, especially for all of you thinking about big data, machine learning, and the potential for this in biology and medicine and any other field, really. Okay. So I want to begin with this quote called by George Spark, which says all models are wrong, okay, but some are useful, and that's the whole part, right? So a critical part of training in doing all this work is not only about the sophistication of these algorithms, but also trying to figure out where they fail, when can they fail, and how do you detect failure, and how do you improve this, and learning biases in data, and so on and so forth. Okay. So to convey this idea clearly, I'm showing you a scatter plot, okay, of x-axis, y-axis. Every single point has an x and y value. Okay. Now, how many of you have made plots like this and fit a regression line through a plot? Okay, some of you have, right? Okay, good. So for example, if I show you something like this, I think many of you might be familiar with what I'm talking about. You fit a line, you can use Excel or Python or R, you fit a line, right? Now, I'm going to tell you that this line is actually a model, just that it's a super simple model. The model is telling you what is the linear relationship between variable 1 and variable 2. And it's actually a predictive model too, which means that in the future, if you give me a value of variable 1, I can predict what the value of variable 2 is likely to be. Okay. Now, even such a simple model can be tripped up by many aspects of the underlying data. So here is an example, right? So somebody in late 1960s published a study where they said there is a huge negative correlation between the number of people who come to vote, this is the voter turnout on the x-axis, and the income inequality in the country, the difference between the richest and the poorest people. And they said that if the income inequality is very high, very few people come to vote, and if not, very large number of people come to vote. Okay, so they published this data, published this conclusion, and then people were sort of taking this seriously and trying to figure out policy. Right? Few years later, somebody actually cared to look at the data and make a plot. Once they made a plot, they found this pattern here. The whole negative relationship, this big, large, thick negative line that they fit, it was hugely influenced by just one data point. And if you sort of ignore this data point, there is almost no correlation between these two variables. Okay, which means that even simple models like this can be very easily tricked by extreme data or hidden data. Okay. Here is another question for you, right? If I told you that the correlation between two variables is almost zero, and if I just told you that statement, in your mind, you might imagine that the relationship between these two variables is like this, right? It's all over the place. There's no relationship. However, if I'm referring to linear correlation, you will still get a value of close to zero, even for data that's like this. Okay. Here, I'm sure all of you will agree that there is clear pattern in the data, just that the pattern cannot be explained by a single line, and hence you will get a correlation of zero. Okay. So it's also important to really understand, if the model is not developed to capture patterns, specific patterns, the model will fail. Okay. And here's a third example. Because all of us are dealing with big data, which means that we have measurements of thousands of tens of thousands of variables over a large period of time, it is very easy to find strong correlations, even when they are real correlations, but the correlation will not mean anything in practical. So here is a correlation across time between the number of movies in which Nicolas Cage participated in, and number of people who drowned in a pool. There is high correlation, but it means nothing. Okay. All right. So here is a summary of many different medical studies put in one case. So I'm sure when you open the newspaper today, in the medical section, I'm sure you read that scientists found drinking coffee makes you live longer or something, right? If you put all these medical articles together, you will see a plot like this, where everything that we eat has been shown to both cause cancer and prevent cancer. So it's kind of ridiculous, right? There are many underlying problems with why we are seeing this. If someone is interested, we can talk along about this. So in fact, so I've seen tweets where Time magazine tweeted, coffee causes is problem, and then coffee helps you live longer. But these two tweets came just 30 minutes apart from each other. Okay, so now the problem is that, why are these problems happening? It is because the traditional statistics courses, the courses that we go to and attend in undergrad and grad school, they actually do not equip us to deal with the real scientific problems, where we have to learn practical definitions of various concepts, not just the formula for a t-test, but what does this practically mean, and what is the relationship between all these quantities? When and why do you use some things and not other things? How do you sequence them? When do you use one analysis after the other? How do you judiciously interpret results, and how do you communicate the findings of analysis carefully to other people, right? So right now, all this is learned by scientists by just talking to each other, talking to their mentors, maybe reading papers, but there is no formal instruction for this. So there is also a lot of intention behind all of this stuff. So which means that lots of scientists come to biostatisticians and data analysts and ask them to do things that are actually not scientifically correct. So this is like, they will say, just alter the data a little bit to support the hypothesis that I already have. Don't report some missing data and so forth. And in fact, all this is relevant to you because all of you are going to do data analysis at some point. And then you are going to get requests from other people. And it is, in fact, frequently reported by younger statisticians that they are pressured to produce positive data, or how they publish totally depends on, what the pressure to publish depends on, completely influences what data they end up reporting. So I've started developing this course called, Gaps, Missteps, and Errors in Data Analysis, to sort of address all these problems. Okay. So this course is very non-traditional. It talks about things that, like, how do you read a paper? What are the biases that both the scientists who wrote this paper have? What are the biases that we have when we read papers? Everything from, things like, how do you create very good data visualizations, and how do you manage code and data and so forth. So my point is that generally these things are important to discuss. Okay. All right. So I want to finish off by giving a huge shout out to everyone in my group, both current and past members. Again, I really want to highlight this. This includes so many undergraduate and high school students who have already worked with us and done some terrific work. Okay. All right. My group is super fun. We love to hang out with each other outside lab, go for movies, go for food, and just hang out. And right now we are in beautiful Colorado. This is the view outside my office. and this is the building, and this is the inside of the building. Anyone wants to come to grad school, please apply to, the graduate programs affiliated with this department at the university. And with that, I want to thank all of the members, funding, and all of you for listening. Okay, I'm happy to take any questions.
Thank you, sir. Thank you very much, sir. It was an exciting talk, and I'm sure it would have made a lot of people curious. Also, truly commendable effort trying to make data available for public. Like a lot of things you have done. I now welcome everyone to ask questions. If you have questions, you can either raise your hand and turn on your mic and ask, or you can just put it out in the chat box. We'll read it for you.
Yeah, Anaga, please go for it.
Yeah, so that was a great talk. I completely relate with the fact that machine learning and such algorithms are not taught in conventional mathematics. Something really interesting about our institute is we actually have an option to choose minors. So currently, I've chosen a data science minor, which allowed me to study machine learning this semester. So this was a
Nice.
particularly exciting talk. One of the questions I had was, um, like as a biologist, I'm familiar with the fact that genes in humans are not studied completely, and across ages, across sexes,
Right.
all that level of data hasn't been studied, and even when studied isn't like readily available.
Right.
So when you're making such functions to for each age you made a different function of genes, how did you process that data? Where was the information from? How was it curated? I was curious.
Right. It's a good question. So, what Kayla, I'm going to describe the work that Kayla did. So, what she did was to look at gene expression data, where every single sample, we know the activity of all the genes in the genome in one sample. And she essentially looked through hundreds of studies and figured out the subset of samples for which people had correctly identified the sex and age information. And she curated all that data. And then the machine learning models, the input is the activity of genes in the genome. If I tell you which genes are active and by how much, can you predict which sex the sample came from or which age group the sample came from? So that's the prediction problem.
Right. And also another question was, I
Sure.
noticed in one of your beginning slides you'd mentioned about model organisms and how I think you can extrapolate it to humans.
Yep. Yep.
So are there any developments in the field that can, go from the mouse model or any other mammal right to humans? Because that's a big question in science.
Yeah. Yeah. This field is very young because there are, including our group, there are probably, you can pick a handful, maybe 10 groups across the world who are really working on this deeply. And so together, I think the field is developing methods to, we are having reasonable amount of success. So I can tell you two things that we can solve in our group right now. So which is that if you gave me a bunch of genes that you found based on a study in humans, we'll be able to figure out how to best map these to the most relevant genes in mouse, the most relevant genes in fish and fly. And this is not just based on sequence similarity. We also use a bunch of other functional data to say that even if these two genes are similar in sequence, they might be doing different functions. It's okay. And then do the same thing back and forth. The other thing that we can do is to say, if somebody, instead of giving us a set of genes, let's say they gave me a transcriptome sample, which is the activity of all the genes in the genome, let's say in a single human patient, we can again map this human patient data to the most appropriate data in mouse, worm, fly, or fish. So we have developed algorithms like this. We are in the process of writing papers and publishing them down the line. So I'm sure you'll see a couple of papers from us, in the next few months.
That's good. Thank you.
All right. Nice questions. Yes. Tasneem, please go ahead.
So in one of the beginning slides, you have shown uh, like you were assigning some values based on the text, just the text available.
That's
So
correct.
how could that be? Yeah. So I didn't understand based on actually what you are assigning those values.
Yeah. Yeah. I mean, this all went pretty fast. So I'm going to, try and maybe spot some of these slides. Yep. All right. So this is probably informative. So The technique, if you want to read up a little bit about this, the technique is called word embedding. Okay, the word embedding idea is the following, right? So our hope is that ultimately we want to turn words into numbers. Okay, we want to represent, like say, hepatocyte not as HEPATOCYTE, but as 9283060, 3267. If you want to get to this, a really good way of doing this is to solve something called the missing word problem, right? So let's say I give you a sentence, and let's say there are 5 words in the sentence, and I deleted the word in the middle. Can you guess what this word is supposed to be based on the 2 words on the left and the 2 words on the right? Right? You can sort of set up this problem for every set of 5 words in an entire corpus of text. So for example, I'm sure you might have heard of Wikipedia or Google News or whatever, right? What if we took every word and every sentence that has ever been written in Wikipedia and then play this game with a neural network model? We say, I'm going to hide the word in the middle, try to guess what this word is, right? And what we are going to ask the neural network to do is to say, in this process of guessing this word, you need to come up with a set of numbers that represents this word so that you can guess the word in the middle. So if you keep challenging the neural network model to solve this problem, what the model will do is to figure out a way to turn words into numbers and predict the word that is missing. Okay. What we do is to complete playing this game and then extract this numerical representations of words that the neural network has learned and use that in subsequent things. I know I'm sort of very hand waving and saying something very abstract, so I strongly encourage you, look up word embedding, and it's a super cool field. You should totally check this out.
Thanks.
Sure. Anyone else? Okay, great.
If there are no further questions, I think we can wind up. I think I'll say I thank everyone who attended this talk and actively participated in the interactive session. I extend my gratitude to our faculty advisor, Dr. Sadananda Singh, and the School of Biology for their continued support towards Proteus. Let's meet on 26 November for an exciting talk by Ms. Madge Britt. Stay tuned. Once again, thank you and have a nice evening.
Fantastic, everyone. See you. Bye.
Bye, sir. Thank you.